Hello, today I would like to talk to you about my recent exploration into the bacteriophage of the female urinary microbiome. First, to better understand the impact of this study, I would like to give some background about the human microbiota. Did you know that you are more microbe than human? The microbiota is a collection of microorganisms, bacteria, fungi, and viruses that inhabit a certain environment. The microbiome refers to the genomes of the microbiota. Thanks to the NIH, extensive research has been done on the overall human microbiome, with large focus on the gut microbiome. However, due to the outdated dogma that urine is sterile, little is known about the urinary microbiome. Over the last 10 years, researchers have found that there are organisms in the urinary microbiome, just at a relatively low biomass. These organisms include several bacteria, such as Lactobacillus, E. coli, Gardnerella, and Streptococcus. And where there are bacteria, there are also bacteriophage, or as we call them, phage. These microscopic organisms far outnumber the bacteria that they infect and play a key role in shaping microbial communities. Phages can predate on bacteria, injecting their DNA into a host, producing progeny, and bursting bacteria cells, thus killing their host. Alternatively, they can integrate into their host genome as latent infections, leaving their host cell unharmed. We decided to begin our survey of the phages within the bladder by first looking at the lysogenic or latent phage population. For this project, 181 bacteria were isolated from 1,000 samples of catheterized urine from female patients. Isolates were sequenced, and their genomes were assembled for downstream analysis. Using a custom pipeline, which uses the tool VirSorter and was developed in Python, I predicted phage sequences within their host genome. Phage genomes were annotated using a program called RAST. Several Python scripts were employed to threshold data and weed out low-confidence predictions. Our analysis identified 457 phage sequences, 226 of which were predicted with high confidence to be complete phage genomes. On the right, we have an image showing the phylogenetic diversity of bacteria found within the bladder. There are many different bacterial species represented in this figure and included in my analysis. On the phylogenetic tree, we have indicated strains containing phage sequences detected by our pipeline. Each colored dot represents one phage sequence. Overall, 86% of the bacteria strains examined by our pipeline contained at least one phage sequence. Some bacterial strains have just one phage sequence, whereas some have multiples. For instance, a Lactobacillus strain has 10 different latent phages embedded in its genome. To delve deeper into the predicted phage sequences, I created a series of Python scripts to determine how similar predicted sequences were to sequences already available on NCBI. This Python script involved invoking the tool BLAST, comparing our predicted sequences to any known bacteriophage sequences. When I compared each of the phage sequences found within the bladder's bacteria, the majority of the sequences were novel, exhibiting no discernible sequence homology to public data repositories. Nevertheless, there are characteristics in these sequences that support our prediction that they are in fact phage sequences and viable. Next, I completed a pairwise analysis in Python, which revealed instances of shared gene content across sequences from different bacterial isolates from different women. One of the instances of shared gene content was in phage genomes found with an Actinomyces species. I developed a script in Python and identified 8 sequences of interest from Actinomyces strains or bacteria closely related to Actinomyces. From this computational analysis, I pinpointed 5 different phage-like regions, shared to varying degrees across the 8 sequences. This analysis shows evidence of a core phagome shared across individuals. This study is the first major exploration of phage within the female urinary microbiome. As such, we uncovered not only a large number of phage, but a high degree of novelty across our results. These results show that while there is a low biomass in the bladder, there is a vast community of phage and evidence of a core phagome in the bladder. While phage impact on human health is known in regards to other parts of the human microbiome, this study shows that there is a need for further investigation into the role phage play in regulating the female urinary microbiota. Thank you for listening to this presentation. Additionally, I would like to thank the National Center for Women in Technology for their consideration for their Collegiate Award.